Hello everyone, my name is Seniha and I will be presenting the paper titled Metabolic Changes in Rat Brain Following Intracerebroventricular Injections of Streptozotocin, a Model of Sporadic Alzheimer's Disease. Here's an overview of my presentation. First, I'll give you some research background, then I'll state the research question. Next, I'll go over the four research methods used in the study and discuss the findings. And finally, I will conclude this presentation with a take-home message. Now, a little background information. Sporadic Alzheimer's disease is a degenerative brain disease that causes a significant memory loss and impairs a person's ability to carry out daily tasks. Abnormal levels of beta amyloid, which is a naturally occurring protein, built up in the brain and form plaques between neurons. Now, another naturally occurring protein called tau collects inside these neurons and creates neurofibrillary tangles or twisted fibers. These toxic buildups of protein prevent neurons from transmitting information, lose connections with other neurons, and die over time. Brain damage related to Alzheimer's disease seems to start at the hippocampus, results in a decrease in cerebral glucose metabolic uptake. Cure isn't found, but there is continued research to find the cause and prevention of the disorder. Streptozotocin, STZ, is a bacterial toxin which damages insulin producing cells and insulin receptors. This was used to inject the experimental groups of rats in the study, which you will see in the upcoming slides. HMR spectroscopy was used to non-invasively obtain a neurochemical profile of rat brains. Research question being, what is the neurochemical profile of animals that received intracerebroventricular or ICV injections of streptozotocin, STZ? Also a statement, quote, the control of brain glucose metabolism is unsettled because of the failure of insulin signal transduction. In other words, the purpose of the study is to quantify the neurochemical profile in the brain area of intracerebroventricular injections of streptozotocin treated rats as compared to the control solvent injected rat. There were four research methods used in the study: animals, 12 Worcester rats, HMR spectroscopy, MRS, magnetic resonance spectra quantification, and gross brain weights. There were 12 adult male Worcester rats weighing from 290 to 330 grams used throughout the study. These rats were housed in individual cages in an air conditioned room with access to water. There were two groups of six rats each that were utilized. The rats in the experimental group received ICV injections of STZ. This group of rats were also anesthetized, while the rats in the control group were solvent injected. They were given ICV injection of the same volume of citrate buffer. These injections were given on the first and third day. The MR measurements were made 3 weeks and 2 months after the first injection, which I will mention later in the research findings section. HMR spectra were measured with the Bruker Advanced 2 spectrometer seen on the slide. A stimulated echo acquisition mode sequence with a very short echo time was applied for localization, and as a result, HMR spectra were obtained from the brain region, including the hippocampus and the cerebral cortex. Using the LC model, which is a linear combination of model spectra of metabolite solutions, there were a total of 19 metabolite concentrations that were quantified as seen on the slide. The data for each metabolite were tested for the normality of the distributions and homogeneity of variances and actually were considered significant. Now, lastly, after the MRI MRS measurement was performed 2 months after the first ICV STZ injection, so the experimental group, the anesthetized rats were transcardially perfused with 5% paraformaldehyde solution. Their brains were removed from their skull to be weighed and stored for further histopathological examinations that were actually not included in the study. The average values metabolite concentrations measured 3 weeks and 2 weeks after the ICV injections are shown in figures 1 and 2 on the slide. Figure 1 on the left shows the concentration of brain metabolites 3 weeks after the injection, and figure 2 on the right shows the concentration of brain metabolites 2 months after the injection. So, in a total analysis, after ICV STZ injections to rats, it produces a range of central effects, including a decrease in brain glucose utilization, an increased release of lactate from the brain, decrease of the level of ATP and phosphocreatine in brain tissues, and disturbances in learning and memory abilities. Now, quick take home message: this study utilized HMR spectroscopy to detect neurochemical changes in the brain region of a small animal model, and after 2 months, is seen that HMRS of rat brains that received ICV STZ injections provides a new input into a better understanding of the critical dependency of neural function and structure on brain glucose consumption. This also may be of relevance to further studies of Alzheimer's disease pathomechanism. Thank you and stay safe.